Everyone. My name is Katherine Sanders, and on behalf of our team, I will be presenting our project titled Healing Effects of Membranes Used for Guided Tissue Regeneration in Immunocompromised Patients. Researchers in this team include myself, Katherine Sanders, Roshni Trivedi, Aditya Deshpande, Connor Schwartz, Dr. Amira Mohajari, and Dr. Manhunk. And for our project, there are no conflicts of interest to disclose. As an introduction to our research, the aim of our project is to investigate the healing effects of membranes used in tissue regeneration in immunocompromised patients. Here is a short overview of some information pertaining to our research project. The goal of tissue regeneration is to repair damaged or lost tissue in the body by stimulating new growth. This method often uses a membrane or a scaffold in assistance. Membrane scaffolds are structures that support and organize biological membranes, which are crucial for maintaining cytoskeletal integrity and function. Types include cytoskeleton, extracellular matrix, and lipid rafts, and each serve distinct roles in cell signaling and adhesion. In our project, we reviewed several conditions that may lead to a compromised immune system, such as osteoporosis, osteopenia, rheumatoid arthritis, diabetes, cancers like multiple myeloma in patients undergoing chemoradiation and therapy, and other HIV-caused immunodeficiencies. Compared to healthy patients, immunocompromised patients have a low bone marrow ability to produce bone cells. Their osteoclast and osteoblast function is affected where the osteoblasts don't build bone as fast as they should. These patients have an increased susceptibility to bone infections, and their bone growth or healing is negatively affected. Moving on, we provided some background information specific to our project. In dentistry, the technique of tissue regeneration is used in many specialties. In our review, we focused on procedures involving periodontal surgery and implant dentistry. Barrier membranes are used as a structural support system that prevents surrounding tissue from invading the site while regeneration of tissue occurs. Membranes are placed between the gingival flap and the defect. This procedure can greatly benefit individuals who suffer from periodontal disease that contributes to bone loss and gum recession. In our project, we were concerned specifically with the healing effects of this procedure in immunocompromised patients. The healing process is delayed greatly in those who have compromised immune systems, depending on the severity of disease. In relation to bone and periodontal tissue regeneration, many complications may arise as the time it takes for the disease to be eradicated in the body is prolonged significantly. There are also many types of human cells that are used in assistance with the membranes. Stem cells are used due to their ability to self-renew and transform into multiple specific tissue cells. For tissue regeneration, these cells are placed on the scaffolds to assist in growth. The articles we have reviewed include mesenchymal stem cells and human periodontal ligament stem cells. The literature search strategy for our project involved finding related articles on scholarly sites such as NIH, PubMed, JAMA, and ScienceDirect. As for the methods used in articles we reviewed, most studies included human cells of different types being injected into immunocompromised mice. Mice are commonly used in research articles because of their genetic similarity to humans, cost effectiveness, and their ease to genetic manipulation. Their short reproduction cycle allows for efficient multi-generational studies, while ethical considerations and the availability of resources contribute to their widespread use in research studies for understanding human diseases. Since our project is still ongoing, we do not have any results yet. However, we do anticipate that there is a significant difference in the hard tissue and periodontal healing process between immunocompromised and healthy patients. We anticipate to find that the time it takes for the disease to be eradicated is prolonged significantly, which increases the risk of further infection in the body. Based off of the articles we have reviewed so far, bone formation was seen to be affected in immunocompromised patients caused by decreased osteoblast function. The studies found that stem cells and tissue-engineered vascular grafts were helpful in increasing bone formation. In the articles reviewed, some of the studies were performed via in vitro and in vivo framework models such as xenografts. However, the majority of the studies that we did review involved human cells. This includes bone marrow cells, periodontal cells, stem cells, and mesenchymal cells. So far with our review, we have found there to be a correlation of materials that were found to become absorbed by membrane scaffolds. Materials such as hyaluronic acid, growth factors, and human proteins that were absorbed by the membrane scaffolds were later determined to have healing properties. In conclusion, there are noteworthy differences in the hard tissue and periodontal healing process between immunocompromised and healthy patients, and the time it took to eliminate disease in an immunocompromised patient's body was prolonged significantly. Bone formation was seen to be affected in immunocompromised patients caused by decreased osteoblast function. Studies found that stem cells and tissue-engineered vascular grafts were helpful in increasing bone formation. Finally, we believe that further research should be conducted to analyze the relationship between periodontal disease and comparing healthy and immunocompromised patients. Last but not least, we would like to thank the Roseman University College of Dental Medicine Clinical Outcomes Research and Education, along with all of the other Roseman faculty that helped us with this research project. Thank you.